Una maestría. Justo hablé con la doctora Eda Shuto el lunes,
Ajá.
porque me gustó un poco su investigación, entonces pues platiqué un poco con ella sobre eso.
Ujú.
Este, y pues, pues estoy pensando, o sea, quiero aplicar la maestría en bioquímica de la UNAM.
Sí, pero eso te va a tocar esperar hasta prácticamente el año entrante, hasta
Sí,
enero del
justo,
año entrante.
porque como me gradué en diciembre, pues me queda entrar en enero, entonces estoy aplicando, se abren las aplicaciones me parece en agosto,
Pero
entonces quiero entrar a ese.
necesitas tener todo. ¿Tú cuándo acabas? Ahora en junio.
Hasta diciembre, mi título sale hasta diciembre.
Mm, está complicado, chavo. Como están ahorita en toda la recepción de papeles del posgrado y todo, este, necesitas tener todo antes de apuntarte como futuro candidato a entrar a la maestría. Y las inscripciones a la maestría para el semestre de diciembre, de enero, que va a empezar en enero del 2026, prácticamente las tienes que hacer, creo que como por agosto o septiembre, y te piden un montón de papeles que ya pretenden que tú tengas. Entonces, ve bien todo eso.
Y sí, bueno, o sea, le estoy pidiendo con un poquito de como consejos y así a la doctora Mara de biomédicas. Y pues de acá de la universidad creo que puedo sacar como papeles, como no el título, pero como pruebas de que ya me voy a graduar. Y como ya estoy en mi último semestre, pues ya puedo sacar todo eso desde el lado de acá de la universidad. Entonces creo que sí se podría. Espero.
No te garantizo. Sí, porque como que últimamente están muy cuadrados en ese sentido. Y
Y
se
sí
ve
está
que,
entonces.
¿sabes? Les han tenido tantos estudiantes que claramente no logran cumplir con toda la burocracia que se les pide, que están, pero así intransigentes, ¿eh?
Sí, sí, imagino. Pero pues sí, o sea, voy a estar tratando de ver, porque sí quiero entrar a la maestría ya en enero.
Mm, muy
Ujú.
bien, bueno,
Sí. Le puse el netido de la plática y está en Week 2025. Ya. Este. Ah, doctora, su presentación se ve como con las notas y todo. No sé si
ah,
lo quiera
se ven las notas y todo?
Sí.
No debería de verse. Está en, este, normalmente no se debería de ver, pero bueno, es tu sistema de...
Yo te recomendaría justo para la maestría, ahorita que estamos aquí platicando tantito.
¿Qué dices?
¿Y usted qué recomendaría para lo de la maestría, ahorita que estamos aquí platicando?
No, pues esa maestría, o sea, es buena, ¿eh? Y y bueno, hay muchos tutores y mucha gente con la que puedes, participar, pero te digo, lo que me temo un poco es que te digan que no tienes todos los papeles.
Sí, que por eso no aceptan.
O sea, que te digan está muy bien, pero a ver si ahora sí ya.
Ahí está, me quedó.
Sí, pues normalmente no se ve lo de los presenter view, pero bueno, no importa.
Sí, no,
Este,
no debería, pero
Eso es lo que me temo, que te digan, sí, bueno, muy bien, aquí lo queremos, pero, y estoy, ¿sabes? Si tienes suerte, te van a dar chance de presentar, o sea, como que van a considerar que tienes suficientes papeles para presentar el examen,
ujú.
y estoy segura que lo vas a aprobar, pero a la hora de la inscripción quieren tener todos los pelos de la burra en la mano.
Y como es la inscripción es después del examen, ¿no?
Sí, entonces, bueno, pues ya verás, pero digo, estate preparado a que te digan que a lo mejor pero si pasas el examen, te dicen, pues ya no hay problema, cuando tenga usted todos sus papeles, el próximo semestre ya se inscribe automáticamente, ya no tienes que volver a presentar el examen.
Ok.
O sea,
Y-
tampoco es que hayas totalmente perdido tu tiempo.
Sí, o sea, se hace la inscripción y a lo mejor no empiezo en enero, pero empiezo en...
No, la inscripción la vas a hacer hasta el semestre de agosto.
Ujú. Ok, ok.
Y bueno, sabes, según si puedes, etcétera, una serie de, ya esos ya son cosas más personales, pues seguramente en el laboratorio de la doctora Shuto te dirán que bienvenido y que empieces a trabajar sobre tu proyecto y que ya luego formalizas la inscripción.
Sí, pues eso podría ser una opción también.
Digo, yo pienso que eso es probablemente lo que podría pasarte, ¿eh? Por los tiempos. Porque digo, están
Ujú.
intransigentes hasta decir basta. En
Entonces sí, a
cuanto,
lo mejor.
en cuanto
Por
a
los...
papel, sabes, vivimos en en el país del papelito habla, entonces,
Sí, como mi título sale como por ahí de enero, o sea, mi título llega a México, sale bien hasta como enero más o menos.
y luego tienes que, sabes, no sé muy bien cómo está ese rollo de que si lo tienes que revalidar, si lo tienes que, es un
No,
título,
tengo un título, o sea, tengo dos títulos, voy a tener un título de Estados Unidos, pero tengo un título mexicano también.
ok,
Entonces ya con ese, voy a, o sea, para UNAM voy a aplicar con mi título de México,
no, pues sí, es lo que te iba a decir.
para hacerlo todo más fácil.
Así, simplifícate la vida, muchachos, si no va a
Sí,
estar.
no, con el otro tener que revalidar y apostillar y
Exacto,
cosas,
tendrías que
no,
hacer un montón de
no, no,
cosas
no.
que no valen la pena.
Mi título mexicano y con eso.
Sí, sí. Sí, no, está bien. Te digo, yo creo que eso es lo que te va a acabar por pasar, ¿eh? Pero tampoco es grave, porque los 6 meses que trabajes, te van a permitir realmente entender mejor tu proyecto, ya realmente estar mucho más a gusto en todo eso, ¿no? Entonces,
Sí, darme un poquito una idea.
sí, no, y no es tiempo perdido, ¿eh?
Ok, ok. Sí, es un tiempo que puedo empezar a adelantar cosas, ¿no?
Totalmente, totalmente
Ok,
puedes hacer eso. O sea, no, no es grave,
ok.
realmente no es grave.
No es grave.
Es
Eh,
más, hasta te podría convenir en el sentido que vas a acabar tu maestría realmente, como dicen ellos, en tiempo y forma.
sí, en los 2 años que son de maestría, ¿no?
Años en vez de los 2 años y medio o 3 que normalmente todo el mundo se tarda.
Sí, porque pues ya aventé como el medio año extra
Exacto,
antes
ya
de
te
la,
aventaste el medio año antes, ¿no?
justo adelante. Pero muchísimas gracias.
Entonces no te preocupes, todo va a ir bien.
Déjenme ver cómo vamos,
Dale,
porque ya tenemos un poquito de gente. Déjenme ver si hay fila afuera y ya para ver. La voy a presentar yo. En cuanto la presente, ya, todo suyo.
vale, vale, vale.
Vale, vale. Déjeme, voy y vuelvo. Vamos en un par de minutos, doctora.
Dale.
Ya también para no tomar tantísimo tiempo.
Sale. Siento, estoy haciendo trampa a mí misma porque para no perder el hilo de mis diapos, estoy haciendo mi PDF y me lo voy a mandar a mi teléfono, porque si no, luego ya no me acuerdo de cuál va. No,
Sí,
sería no
claro que sí.
acordarme exactamente. Y hay cosas que no quiero explicar con mucho detalle porque no viene al caso.
Sí, pues sí, para no confundir tanto a los chicos que a lo mejor no tienen tanto background como en inmunología y así, ¿no?
Exacto, sí, exacto. Entonces nada más quiero pasar como muy rápidamente sobre de ellas.
Sí, va que va. Déjeme mandar un mensaje rápido y ya.
Vale, dame un segundo más para que acabe yo de pasármelo a mi teléfono.
Por supuesto.
Listo. ¿Y tú tomaste un curso de inmunología?
Estoy trabajando con la doctora Nora y
Ah,
fue
estás con
mi...
Nora. Ok,
Hice mi servicio social con Nora todo un verano. Fue mi crash course
ok.
de inmunología y aquí en la
Ah,
universidad con una doctora tomé dos materias como inmunología general y inmunología avanzada.
pues esto no está mal. No está mal.
Sí, entonces, sí, la verdad es que ahí me decidí por mi maestría. Quiero avanzar el enfoque en inmuno justamente.
Sabes, Nora fue una de mis estudiantes de doctorado.
¿En serio?
Sí. Ah, y el doctor Aleph también.
¿También el doctor Ale fue estudiante de doctorado?
No, él fue de maestría,
¿O el
pero
estudiante de maestría?
sí, sí, sí.
Siempre siento que, bueno, me encanta porque todos entre ciencias como que sé que nos conocemos entre todos.
Sí, nos conocemos entre todos, ¿sabes? Y la verdad es que sí, como que llega un momento en que dices, bueno, pues sí he hecho
Como mucha
mucha,
conexión.
mucha contribución a formar recursos humanos.
Y sí, por ejemplo, de hecho también el esposo de la doctora Shuto es amigo de la doctora, creo que Ileana Barrera, que invitamos, que está en CIDETEC, y trabajamos juntos en un consejo durante un tiempo y así, entonces nos estuvo contando ayer.
Ah, sí, ya
Sí,
ves.
justo.
Entonces sí, no está todo. O sea, tampoco es que seamos muchos, ¿no? Entonces, al final de cuentas,
También, sí, sí. Ya está listo, doctora, cuando usted me diga.
cuando tú quieras.
Entonces ya nada más me acomodo el micrófono y ya estamos. ¿Ahí me escucha bien?
¿Sí?
Perfecto. Entonces vamos a comenzar. La voy a presentar.
Pues ya están llenando, pero no demasiado, ¿eh? Como siempre, todas las hileras del principio están vacías, ¿verdad?
Voy a pasar ya el audio de la computadora hacia el salón.
Órale, entonces ya nos callamos, ya no hacemos comentarios.
Listo. Listo, así acabamos. Okay, hello everyone, and thank you so much for joining us today for the first talk of the third day of this STEM week. To start the day off, we have Dr. Yvonne Ronsenstein. She was trained as a biologist at UNAM, and after earning her PhD at Sorbonne University in Paris, she worked as a postdoctoral fellow and assistant professor at the Dana-Farber Cancer Institute at Harvard University in Boston. Currently, she's a professor at UNAM's Biotechnology Institute in Cuernavaca. She and her research group are focusing on the molecular mechanisms regulating immune signaling, primarily how co-receptors like CD43 initiate various signaling pathways that modulate antigen receptor signaling, influencing T cell activation and decision making. They now utilize animal models to investigate its role in immune cell functions, as CD43 interacts with pathogen-derived molecules such as M. tuberculosis and S. gordoni. Furthermore, they aim to discover novel immunomodulatory host defense peptides to develop treatments for chronic inflammatory and infectious diseases. They established a pipeline to test three frog-derived peptides, taking candidates with promising immunomodulatory properties as opportunities for innovative therapeutic approaches to chronic inflammation. In addition to her research, she serves on several academic committees, and she's currently the president of the Mexican Society of Immunology. Please welcome Dr. Ronsenstein with a round of applause.
Well, good morning, everyone. First of all, I would like to apologize for not being able to be with you today, but I wasn't able to manage and get to Querétaro and Juriquilla and do every other things that I have to do here in Cuernavaca. And I also want to thank Sebastian for inviting me. He already invited me last year, but I wasn't able to be there with you. And so this time when he invited me again, I decided that this time I really wanted to be there. So today I'm going to talk to you about what we do in immunology research. And the title of my talk is Exploring the Immune System Through the Lens of CD43, which is also called cyanoferine. So my talk would be divided sort of in 3 or 4 um, parts. I'll talk to you about what is immunity. I will talk to you about what is the immune response and how does it work. What we do in our laboratory so that you have an idea of what immunologists do in their labs. And then we would do some concluding remarks about all what I have told you. And I hope that I am able to transmit you in a simple language everything, all those concepts that tend to be sort of complicated. So what is immunity? Nowadays, when we talk about immunity, we think, of course, about the immune system. However, believe it or not, it was Thucydides, which was a Greek philosopher, who was the first to translate the term of immunity to diseases, because immunity means exception, privilege, of license. For example, you are exempt of paying taxes. That's what immunity means. Or you are exempt, or you are you have the privilege of not being in jail if you are the king, for example. So, and he did that because there was a huge epidemics of plagues in Athens in 430 before Christ, and nobody knows exactly what that plague was, but nevertheless, 25% of the population of the city of Athens was killed during that time. And it was the first time that immunity was used, the term of immunity was used to describe those people that didn't die during the epidemics. And so they were immune, they were exempted of the disease, and they survived. And that's why immunity became the sort of the equivalent of resistance to disease. And then, and you will see why I'm talking about those two guys, and Hippocrates, who was a very skilled philosopher and doctor also during the Greek ages. He said that the disease results from an imbalance between the body's 4 humors. So you have to have all those 4 humors, blood, what he called blood, black bile, yellow bile, and phlegm, which had to be in balance for you to be healthy. If they were in imbalance, you would be ill. And so what were the elements that will be important for that? And so he thought, he didn't have any real proof, but he thought by his experience, by treating all the people that he, as a doctor, had to see, he thought that the environmental, the dietary, and the lifestyle were absolutely important to regulate that internal balance between the body's humors. And then he also knew, because of what he observed, that individuals healed when the balance between the 4 humors were restored. So we're talking about a concept that now we know as homeostasis. So how does the immune system work? Okay, so based on what I told you, the immune system is a system that is capable of recognizing, detecting potential threats. And so you have cells in tissues. Let me see if I can put my laser pointer. Do you see my pointer? No, huh? Oh, yeah. So you have, so this is the yellow cloud, the pink cloud is a tissue, and in the tissue you have healthy cells, and you can have also a cell that is sick. And it's sick because it has some abnormal, it's telling the environment, it's telling that it has been attacked by a virus or a bacteria. And what happens there? In those tissues, you have also local immune cells that will live in those tissues and will detect the threat on that sick cell. not an abnormal cell. And those local immune cells, what do? Then what they do is that they secrete chemokines, which are sort of hormones, if you like to talk about like that. And those chemokines will diffuse and they will attract other cells to the tissue that is being attacked by the pathogen. So we have all those cells that will get there. I can't see my pointer. Oh, here it is. So you have all those cells that arrive there and they say, we've got chemokine signals and we're here to help. And that's how we get inflammation, swelling, and we get all these cells that get into the tissue to attack the germ or the cell that is being sick. And by the end, if they do that in a balanced way, then we will heal, and all the equilibrium, the homeostasis, will be regained again. So I already told you that the cells of the immune system arrive, and they arrive because the immune system is not localized in one single organ. The immune system is very diffuse. It's all the cells from the immune system arise from the bone marrow, but then we have in our body what we call lymph nodes and secondary lymphoid organs that are where the cells the cells will get into all those different organs, secondary organs like the spleen, like the lymph nodes that we have all around our body, and they will do a surveillance system where the cells are informed about what happens in the periphery, what happens in those tissues. So if you have a threat in one tissue, and then you have lymphs. You can see that we have lymph nodes all over the body. So you have cells from those tissues that may travel from the tissues that are being affected to the closest lymph nodes. And there in the closest lymph nodes, all the cells of the immune system can get in and out, and they get in and they talk with those cells that are bringing the information from the tissues that are threatened by the germs. And so they will know, they will get the information and they will get activated. So what happens? So by the end, we have two sort of speeds in terms of how we deal with infections or with diseases. We have the very fast speed, which is our innate immune system, and it's in that innate immune system, we have a bunch of cells that participate, and that many of them are tissue residents. That means they are in the organs, in the different organs that make up our body. And from there, they will deal with the infection. And then we have the more slow immune response, which is the innate immune response, and where we have all the cells, and we will talk about this later on, and those are B cells and T cells, but I'm not going to talk more about that. So we have two speeds. And we have two speeds and we have different cells. And we have all those cells of the innate immune system that will sort of eat very fast. And those are macrophages and mostly neutrophils that have the capacity to eat all the invading pathogens, and they have the capacity of cleaning the site. But however, if they cannot do that, then they will secrete all those cytokines and chemokines that I talked to you a few slides ago, and they will call other cells to come, and they will call the cells of the innate immune response. So by the end, in the innate immune response, we have T cells that are the blue ones, and we have the gray ones. We have T cells, and we have B cells that make antibodies, and we have cells that kill, and we have cells that kill other cells. So the T cells, we have different kinds of T cells. They can be either CD4s or CD8s, and they secrete a lot of cytokines and chemokines, and with all those cytokines and chemokines, they will orchestrate everything that happens in the lymph nodes and everything that happens in the tissues that are being threatened by the pathogen. The B cells will secrete antibodies, and antibodies, you all know now what antibodies are, because we have been, most of us, vaccinated against COVID, about 3 years ago, 4 years ago, and we have learned that we had to make antibodies against the COVID-19 virus, right? The SARS-CoV-2 virus. And then we have those cells, there are T cells also, that have the ability of killing those infected cells. So you have a team of cells that will do either the very fast response or the very slow response. So how do we study all those cells? How do we know all of that? So I'm not going to talk to you about general things, but I will talk to you about what we do in our lab to understand the mechanisms that govern T cell function. Because in our lab, we mostly have been studying T cells, but we are now doing other things, as I will tell you by the end of my talk. So we know now that you need at least several signals to activate a T cell. You need a signal that the T cell has to recognize the pathogen, and it does it so through a special structure which is called the receptor, the T cell receptor, and the T cell receptor recognizes not the whole pathogen, but just a tiny bit of it, which is called a peptide, and that peptide is presented to the T cell through a specialized cell, which is called an antigen presenting cell. So you need the signaling of the T cell because the T cell receptor has an intracellular, it's a protein on the cell surface, and it has a signaling machinery that allows the T cell to know that the T cell receptor is being activated because it recognizes its peptide, its antigen. But that's not enough for the cell to be activated. The cell needs to be absolutely sure that it will go into a process of activation, which will lead him to proliferation and then differentiation and cytokine production. So, because you have to know that once you get a T cell that is activated against one peptide, one molecule, it will take a very difficult decision, which is, shall I proliferate? So that means that from one cell that recognizes one protein, you will have an army of one or several millions of cells. So you can envision that this is a huge challenge. It's a huge challenge in terms of energy for the body. So that's why when you get sick, you feel and you haven't been able through your innate immune system to deal with the pathogen, then you really start to get sick and to feel that you need to stay in your bed. And you need to stay in your bed because you need to derive all this energy that it takes for the cells to decide whether they will proliferate. And you will do from one cell several millions of cells. So that means you need to stay in bed and save your energy for that. So to do that, to take that big decision, you need at least a secondary signal, which is a signal provided by what we call in immunology co-receptor molecules. So those co-receptor molecules will complement the signals that the T cell receptor will give to the T cell. And so they will add up. And by the end, as you can imagine, a cell has thousands of molecules on its surface, and all those molecules have different functions. So by the end, the lymphocyte, the T cell, integrates all the signals that are provided by all those molecules on the cell surface, and they integrate that into multiple signals, and they take the decision to proliferate, differentiate, and produce all those cytokines or all those enzymes needed to kill the cells that are infected and that are sick, so that by the end, the immune system can deal with the pathogen that is attacking you. So the molecule that we study in our lab, it's one co-receptor molecule that we call CD43. Here in this cartoon, I've showed you that so that you can see that we have at the end, we have the T cell receptor that recognizes the peptide presented by the antigen presenting cell on blue. And then you see that there is a lot of other molecules and a lot of co-receptor molecules. I've seen in the red circle, you have CD28, which is the one molecule that I was showing in the previous slide. And then here we have CD43, which is a big molecule. It's the first interesting thing about CD43 is that you have about 500,000 of those molecules on the surface of a T cell. So it's very, very abundant. It's about 10 times more abundant than any other molecule on the cell. It's present on all lymphoid cells, so it's an important molecule. It's present from the onset. It's not something that appears on the cell surface as a result of the activation that the cell undergoes. And it has no enzymatic activity. It's a long molecule and rigid molecule. It's here on the, you have the extracellular space, and you see that it's a very glycosylated molecule. So it's a very what biochemists will call a sticky molecule, and it's a very long one and a very rigid one. It's much longer than many other molecules. It's about 3 or 4 times as long as most of the molecules on the cell surface. And its intracellular region has a proline-rich region, which is important to signal. It has several regions that are important for the cells for the molecule to signal, to transmit signals into the cell surface. Once by the extracellular domain, it recognizes its ligands. So as I told you, it's a glycosylated molecule. And so those molecules are usually very promiscuous, and they have many functions. Promiscuous means that it has a lot of ligands. And see, you can see on the side of the screen that CD43 has many ligands. It has ligands that are, let's say, physiological ligands, and it has ligands that are also ligands that are found on pathogens like Trypanosoma cruzi, Mycobacterium tuberculosis, Streptococcus gordoni. The HIV patients have anti-CD43 antibodies. And which is interesting is that all the physiological ligands that CD43 has are also shared with some other molecules. So it's clearly not a molecule that has one ligand. It has many ligands. When it interacts with each of its ligands, it probably transmits different signals to the cells, but that we don't know very well. We know very much about many of the signals that the CD43 transduces that I will talk to you in a few minutes, but we don't know exactly which ones are transduced with each one of those ligands. And so, because it has so many ligands, it participates in many functions that are important for T cells to do their function in the body. So they participate in adhesion, they participate in regulating cell survival, which is important for a cell, and they participate in inflammation and proliferation. So, and those are the points. So one of the first things that we decided to do in our lab was to, considering that CD43 is such a long molecule that it protrudes, that it's so much away from the cell surface, does it function like an antenna for the cell? And so what we did was to activate the cells with the T cell receptor and then with CD43. But what we did was to do that at different times. So we first did CD43 and the TCR together, or we did CD43 for 2 hours and then the T cell receptor activation, and then the T cell receptor or first and then CD43. And we did that with T cells that we isolated from blood bank donors. When you need lymphocytes and you work in a lab, in an immunology lab, it is very frequent that you go to the blood bank and ask them to give you what is called a leukopaks. Leukopaks are just what the blood banks don't use, because those are the lymphocytes that they take from when you go to the blood bank and give blood for transfusions, the leukocytes are the one part of the blood that they cannot use. So in the labs, we can use them. And so we got there and asked them for some leukopaks. And we isolated the T cells from those donors. And we activated, as I just told you, first CD43 and then the TCR, or the TCR and then CD43. And we asked several questions. We asked, are those cells going to be able to proliferate? Are those cells going to be able to secrete cytokines? And what are the biochemical mechanisms that are involved into those responses? And if I can change, oops, I think I have a problem. There. And so the first thing that we were able to see, was that when we activated the cells with either CD43 first and then OKT3, or if we activated them together, the cells would proliferate. With OKT3 means the T cell receptor, okay? I'm sorry. And whenever you see L10 means an anti-CD43 antibody. So we activated the cells by adding those antibodies against those two molecules, and that's how we activate them. And so we mimic the interaction of the molecules with their ligands. So when we activated the cells first with CD43 and then the TCR or together, we have a big response. The cells proliferate. But that was not the case when we activated them with the T cell receptor and then with CD43. And the same thing in terms of cytokine production, what we did was to see whether those cells will produce an interleukin, which is very important for cells to proliferate, which is interleukin-2, which is called interleukin, but it's in fact a growth factor that the cells need to proliferate. And so we found that when the cells were activated with first CD43 and then the T cell receptor, they produced large amounts of interleukin-2, the same as when the cells were activated with the T cell receptor and CD43 at the same time, but not at all when the cells were activated with the T cell receptor first and then OKT3 and then the T cell receptor. So that meant that the sequence is with which a cell recognize or receive the signals from the different molecules that participate in T cell activation is important. The sequence matters. And so from a biochemical point of view, I don't want to bother you too much, but what we were able to find was that when you activate with first CD43 and then the T cell receptor, you see that the activation state of this molecule that we call ERK, which is a kinase that is important for the cells to proliferate, the activation of this molecule is very long and very much more intense than when you have, for example, the cells that are activated first through the T cell receptor and then through CD43. So you see that the pattern is completely different. And we were able to find other biochemical cues that allowed us to understand how those things happen. And if you do that, if you put all of those molecular evidences, we were able to demonstrate that when the cells are activated through CD43 first or at the same time as the T cell receptor, then you have a signaling cascade that is enabled and reinforced, and that goes through precisely this molecule that we call ERK, this kinase, and that will lead to the proliferation of the cells. However, if we activated the cells with the T cell receptor first and then with CD43, we had the expression, we had the ERK was not activated for such a long time. That enabled the cells to recruit a phosphatase that will then block the signaling very early on at the level of the T cell receptor, and the cells will not proliferate, and that will be those cells will be sort of what we call in immunology anergic. So that means those cells generate a lot of intracellular signaling not to be able to respond. And so We also wanted to know whether those cells that were activated with different ways and where they perceived the signals of this long molecule that we call CD43 would secrete different amounts of different cytokines, depending on the order where the molecules were called to participate. I already told you that the cells produced when they were activated through CD43 before or at the same time as the TCR, the cells produce large amounts of interleukin 2, but we want to know whether they would secrete other cytokines. And, in immunology, as I told you, and as Hippocrates already envisioned, it is all in the balance. All the humors, as he called them, had to be balanced. Because if when they are not when they are in balance, we have disorders. We have different kind of disorders. I just want you to look at this just for a second because I will come back to this later on. We have different disorders depending on whether we have too many interleukins of what we inflammatory interleukins or too many anti-inflammatory interleukins, and that will give rise to different imbalances and will give different diseases. So I just want you to look at how different the production of different cytokines can be, whether you activate the cells with the T cell receptor and CD43 at the same time, or CD43 before. You see that all these little black dots are all the cytokines that the cells secrete in response to those signals. After those were the same T lymphocytes that we isolated from the blood bank donors, and where we did the biochemistry, we also recovered the supernatants that we have, the liquids where the cells were incubated during the activation period, and we asked whether those the cells had secreted all those different cytokines. And so you see that they do a lot of cytokines and they secrete a lot of those. And however, when the cells were activated with the T cell receptor first and then CD43, you remember that from the biochemical point of view, I showed you that the signaling in the cell was not the same one, that it had it was blocked at the level of ERK. And because of that happened, the cells were not able to proliferate, but they were not able to produce interleukin 2, and they were not able to produce either any of those cytokines that the cells produced when they were activated together with the T cell receptor and CD43, or with this molecule CD43 and then the T cell receptor. You see the landscape is totally different. And that you needed the collaboration between the T cell receptor and this molecule CD43 for the cells to really get truly activated and proliferate and secrete all those cytokines. Because when you see what the cells were activated either with CD43 or the T cell receptor alone, they produce very few cytokines. So you need to know that those cells are really integrating the signaling from all those molecules at the same time. And by the end, it's like a huge computer. And by the end, they decide either to proliferate, to differentiate, and to secrete all those cytokines, or they don't do nothing, except that they secrete a few cytokines. But like, for example, an interleukin that is called IL-8. And IL-8 is very important because it's what could be called like an alarmin. It's something that will tell the cell, the organism, that it's a cytokine as well, and it will tell the organism that something is wrong and that something has to be done, even though that cell cannot do it. So to make you a long story short, we went on asking those cells what happened when you activate the cells through CD43 and then the T cell receptor. And we found that we had a certain number of processes by doing a proteomic study when we already knew that we had all those signals leading to ERK activation, but wanted to know more. So we decided to activate the cells just as I told you before, and to lyse them and take them to do a proteomic analysis and see what proteins were overexpressed or phosphorylated. And what we found out is that we have a certain number of biological processes that were activated, that were regulated. So we found that we have metabolic processes, that we have apoptosis that was regulated, and that we have, of course, a lot of regulation of different metabolic processes that were involved in T cell proliferation. And I don't want to bother you and have you understand all of what I'm telling you, but biochemical techniques, we were able to demonstrate that indeed, when we activated the cells through CD43 and then the T cell receptor, we would activate the cells. We would activate the cells through that we will inhibit the apoptosis. Apoptosis is a way of that the cells die, which is very important in immunology. Immune cells tend to die by a process that is called then apoptosis, and it's regulated by a certain number of molecules. And so we were able to show that we had molecules that were that will inhibit apoptosis, like CREB, and like BAD, that would their expression would prevent the cells to get into apoptosis. Well, BAD actually was diminished, and that we had molecules that will promote cell survival. So by the end, we have now a very complex image of all the signaling that is regulated through CD43. And the take-home message is that CD43 signaling parallels very much the T cell receptor signaling and reinforces it, which is very important for the cells to take the decision to proliferate and to produce certain cytokines and chemokines. And as I just told you, the CD43 signals will promote cell survival. So how did we do that? So how did we know that? What we did was to generate cells in the lab that would express either a wild type form of CD43 or a form of CD43 that expressed that lacked the intracellular region of CD43. If it didn't have the intracellular region of CD43, then CD43 would not be able to signal because it was a truncated form, okay? And so what we found, so that was cells that were activated through PMA and ionomycin, which is a very strong stimulus. When the cells were activated with PMA and ionomycin, the cells that express the wild type form of CD43 would die up to a certain point. Yes, because that stimulus is very strong, and and after a certain point, the cells had proliferated so much in the wells that they started dying because they didn't have any more nutrients in there. But the cells that lacked the intracellular region of CD43 died much more, as you can see. So by the end, what did I tell you? I talked to you about this co-receptor molecule, and I showed you that CD43 serves as a co-receptor. It completes, it integrates with the T cell receptor signaling, and CD43 signaling through CD43 helps lower the threshold for T cell activation. You need less T cell activation, TCR activation, and then you have all those signalings provided by those co-receptor molecules such as CD43, but there are many more other molecules on the T cell surface and on many other cell surfaces that will do the same thing. They will help lowering the threshold for T cell activation. The T cells will be more fitted to get activated, and if they have the right amount of nutrients in the medium, they will go into proliferation, and they will go into differentiation, and they will secrete different kinds of cytokines and molecules that will orchestrate the T cell, the immune response. And so also, which is important in terms of immunology, is that CD43 initiates signaling pathways that direct T cells toward various phenotypes. And I wrote here primarily Th1. What does that mean? Well, when you have an immune response, you can have different scenarios. And the best of the scenarios is that you have enough of everything to deal with the pathogen that you're dealing with. And which is not, and so that enough of everything means at some point it can be, you need a lot of cytokines, like will promote inflammation, like what we call the Th1, that promote a Th1 response, and it's an inflammatory response, and that's important at the beginning of an inflammation, of an infection. And that what CD43 does, it differentiates the cells towards a Th1 phenotype, which is important. It might not be a good thing if it's too much of that. And then I showed you that the order of the factors change the product, and which is important. So when you have a cell that have so many molecules on the cell surface, you can envision several scenarios again. Does the cell signal through all of those molecules at the same time, or it doesn't? It's possible that both things may happen. In some cases, you have molecules that are so long, like for example, CD43, that is so abundant, that has so many ligands, that it will interact with some of its ligands at some point in different regions of the body, and that will start signaling into the into the lymphocyte, and it will start preparing it for different functions. It is also possible that the T cell receptor encounters its antigen first than any other molecule encounters its ligand on the antigen presenting cell, and that will lead to a different and interpretation of those signals. So the order of the factors can change the product, and that's important. And another aspect that I haven't talked to you about is whether CD43 has a role in cellular transformation. Cellular transformation is what in science it's a prelude to calling about a cellular gets transformed and then it gets cancerous. It gets a lot of, so transformed cells are cells that are precancerous, and CD43, and I haven't showed you that, and I will not today, but participates in cellular transformation at different levels. So we have there a possibility of, we have identified a molecule that will participate in different functions for T cells, but that will also be important because those cells, when they are expressed by tumor cells, then they can also signal into the tumor cell and provide the tumor cell some advantages. And that's what CD43 does. So to come back to the beginning of my talk, what does the immune response do? Well, it's a system that enables you to control different threats. It can be threats like tumor cells, like foreign particles, like viruses, bacteria. And it will allow you, if everything goes well, to respond to those threats and eliminate them, either through your innate immune response or either through your adaptive immune response. You will respond by secreting cytokines, inflammation, and you will activate all those cells that will either phagocytize, eat all those bacteria, the foreign particles that have invading you, all those cells that are infected by viruses or those tumor cells. And you will have antibodies that will help you to neutralize all those threats. But the immune response can go wrong too. So you can have either you can develop what we call now an autoimmune response, which means usually as an individual you preserve your identity by not recognizing yourself as a foreign matter, right? Your immune system is made in such a way that it recognizes that you is you. And so that's the way you can recognize that something different to you, to yourself, is not you. So, but sometimes it gets wrong, it gets confused. And so it recognizes itself and you get an immune response that is directed against the body. And that can, of course, because then everything happens, the same thing as it happens when you have a bacterial infection, then the T cells and the innate lymphocytes and the B cells, everyone gets activated, and you get an immune response against yourself. And you have different diseases like multiple sclerosis, like lupus, like rheumatoid arthritis, that are what we call now an autoimmune response. And then you have also when your immune response is so good that it is exaggerated. There is not that balance that Hippocrates talked about already. It's unbalanced. And so you have immune responses that are so exaggerated and that cannot be controlled by the body. And also you have those responses that will damage your healthy tissues. The same way as the autoimmune response will damage your healthy tissues, the exaggerated immune response, like allergies, like asthma, like inflammatory bowel disease, or a response to alloimmunization, will lead to an exaggerated response, and it will also damage your tissues. And then you have sometimes nothing happens. So that's unfortunately that's what happens when you have, for example, a cancer. Your cancer cells are smart enough to bypass the immune system, or to tell the immune system that, yes, you know that I'm here, but never mind, we're friends. And so by the end, the cancer overgrows and your immune system cannot control it anymore. And so you can sort of say that the immune response is ineffective. It fails to recognize and to respond to the threat. So, but now I just wanted to show you this picture very fast and to show you how we have been, but maybe I will go first to this one. Sorry. Um, the immune system is just done much more than protection against infectious agents. Hippocrates already said it before, many, many years ago. And it was again, this idea was again taken back by a French physiologist, which is called Claude Bernard, and he talked about the milieu intérieur. And the milieu intérieur is all this homeostatic milieu where everything in your body is in balance, as Hippocrates said. Everything is balanced at the right size, the right proportions, so that all your organs work in harmony and your body functions well. And the immune response is in the middle of all of that. For many, many years, we thought that the immune response was just responsible for protecting us against infectious diseases. And then we understood that we could manipulate infectious disease and develop vaccines. And then we understood that also by manipulating the immune response, we would be able to manipulate and to make transplantation successful. Transplantation, we transplant lungs nowadays, we transplant kidneys, we transplant hearts, and because we are able to manipulate the immune response, we also were able to learn that immunology is at the center of how our body is able to deal with cancer cells and tumors. We are now, unfortunately, we are also now recognizing how important is the immune response in autoimmunity, in inflammation, and in general in inflammation. And so by manipulating the immune response, we're now able to do what is called a precision medicine. We can use immunological weapons, to talk about that like that, to make tissue repair, to treat cardiology, arteriosclerosis, to favor wound healing, or to understand better neurological diseases and neuroinflammatory diseases. And if we are understanding the role of the immune response in all those diseases, we are now able, because we are able to manipulate the immune response, we are able to generate cells that are very engineered in terms of that they can express different molecules on their cell surface. We have we can make them to express specific receptors for certain antigens. And so we can use immunology as a weapon to augment our resistance to diseases, to recover from many diseases, and then to maintain our health. And then I will go back to our very last, to this last slide, or very almost last slide, and to show you, if you see, it took us a long, long time to go from the first vaccination by Jenner in the 1700s. That was the first vaccine against smallpox. And then if you see how the intervals between the dates get smaller and smaller and smaller, the importance of immunology can be understood, or has been understood, by the number of Nobel Prizes that have been given to important immunologists that have participated into the discoveries, and to show how important is immunology in terms of understanding how we can improve the health of people. And so, and that will give you, so you see the span of time is shorter and shorter and shorter. And then to culminate with the last Nobel Prize that we had to those two individuals, Kathleen Calico and Irving Weidman, to that developed the COVID vaccine that we were all benefited from. And with this, I would finish my talk. And if I was able to stimulate you and to make you, for those of you who are not close to immunology, if you are interested in getting, okay, what do immunologists do? So I will invite you to go to the Mexican Society of Immunology, La Sociedad Mexicana de Inmunología, and we publish now a small divulgation journal that is called El Infocito. And when we have different articles, and where we explain in simple terms what immunology is about, and we hope that we can interest you with that. With this being said, I would tell you that, this is my group, that was last week, and we were celebrating Luis Chipre's graduation. He finished his master's degree, and so we were there all just after his presentation. And I also would like to thank mostly all my collaborators and also all our funding agencies, because all of what we do is made possible because we get funds from the UNAM, from the CONACYT, and from different from different sources. So from this, I'm happy to take questions, and I hope you will have questions.
All right. Thank you so much, Dr. Ronsenstein, for this amazing presentation. Do we have any questions? Yes.
Hello. Thank you for your talk. It was very interesting. I really like it, and I wanted to know, like, what were the challenges that you faced during the all this research of your project?
Well, let me tell you, the first challenge is having enough funds to pursue your research. So we have to really look for funds in our labs, and so that takes a lot of our time, actually. And once you get there, what are your challenges? Your challenge is to do the best science you can with what you have, okay? And to understand, to generate a question that really will respond or will complete the actual knowledge that you have. And why is it that important? Because what you want is to contribute to the knowledge, to the general knowledge. You want to bring your little grain of sand to the general knowledge. And by doing that, what you will do is to allow other people to go from what you have discovered and go further ahead. And so the real challenge here is to, when in universities, what we do, we train students, and we do, of course, our research, and we have this area of research that interests us. But then we have young students like you that come into our labs, and the biggest challenge is to introduce them to the discipline that we're working on, and to give them the best education we can, which means we have to tell them or discuss with them, because we talk a lot with our students, and then we have to take them through how do you do work, how do you work, what do you do to make sure that your results are reproducible, what do you do to make sure that you're working with certain standards, and what happens if you're not, and how can you learn those skills. So maybe the largest challenges is to talk with our students and make them great researchers. So see, through my career, I have had the opportunity to train quite a lot of students, and I'm very proud because many of them are now very skilled researchers, and they are in many different universities in Mexico and sometimes outside of Mexico. So that's my biggest challenge and my biggest satisfaction, I would say.
Thank you so much.
Thank you. We have time for one more question.
Hi, Dr. Rosenstein. That was a beautiful talk with a lot of very nice diagrams for people who are not very acquainted with immunology. So thank you so much. I wanted to ask you if you know if there's anybody trying to take a look as to the activation of this molecule in patients who have reactivation cases of tuberculosis, in whom we really want to skew the immune response towards this Th1 phenotype. And it is increasingly difficult when they have had the infection for quite so long.
No, but your question, thank you so much for your question, because that will, see, we have, I didn't talk to you, but we have a huge project on evaluating the role of CD43 in tuberculosis. So we developed a model where we have wild type mice and mice that are lacking the expression of CD43, and we have infected them with tuberculosis. And what we have noticed is, curiously enough, I mean, which is CD43 participates in the infection of the macrophages, which are the most preferred cells for Mycobacterium tuberculosis to infect. And so if you are lacking CD43, then the cell does not get so much infected. But curiously enough, those mice that are CD43, that are lacking the expression of CD43, they are less infected at the beginning, but by the end, after 4 months of infection, they have more bacteria in their lungs than the wild type mice. But curiously enough, they die less than the mice that have CD43. So that means that CD43 participates at two levels of the disease. First, at the entry of the bacteria into the cells, and then also in the defense of the organism against the bacteria. So, and because of the data that I showed you, that CD43 polarizes the cells towards a Th1 response. So if in those mice, if you lacked the expression of those cytokines that promote inflammation, as the Th1 cytokines do, then even if the mice has more bacteria in its lung, it's less inflamed. And because it is less inflamed, it has less destruction of the lungs by the end. And by the end, the animals live longer. So, and then the response is no, nobody has really paid attention to that. And so we are about to publish those results, and we think they are quite interesting in terms of that disease in particular. And thank you so much for the question.
No, thank you. That's very interesting. Thank you, Dr. Rosenstein.
Right. Thank you so much for your time, Dr. Rosenstein. Can we please give her a round of applause?
Well, thank you so much. It was a pleasure, and I would have liked to be there, but I wasn't able to be there.
Thank you so much, Doctor. We'll be sending your certificate through email later. We want to invite you all to stay for our next talk, which is happening right now with Dr. Erika Bustos. So please, we invite you to stay here. And thank you so much.
Thank you.